Welcome to the botany class. Students, I am Dr. Pramod Khadse, Department of Botany, Sri RT College of Science, Akola. Today we have discussed or study regarding the Hatch Slack pathway or the C4 pathway. So here, student friends, Hatch Slack, he was a well-known scientist and he discovered this pathway in sugarcane. The first products are generated in this pathway is a C that is made up of four carbon compounds, dicarboxylic acid compounds. That's why the name of this pathway again it is called as the C4 pathway. And due to the investigating this sequence of this reaction in this pathway by these Hatch and Slack, that is why again the name of this scientist are given to this pathway, Hatch Slack pathway or C4 pathway. Generally, this pathway is found in generally in the plants that is sugarcane. But in many plant species of the family Gramini, for example, the maize, sorghum, which is a group together as called as the tropical grasses, and other name of these plants called as the uh, other example of these plants that is the Atriplex and Amaranthus. So these are all known as C4 plants, and it is distinguished by absence of photorespirations and the anatomical similarities of the leaf, that is the cane type. But here the anatomy of this leaf is very, that is interesting and very characteristic. Here why? In the leaves of these plants the vascular bundles are surrounded by a bundle sheath, that is a bundle sheath of larger parenchymatous cells, which in turn are surrounded by a smaller mesophyll cells. So here in the leaves of these plants the chloroplast in cells of bundle sheaths are larger and usually lack the grana, but the chloroplast in mesophyll cells are smaller and always contain granum. In the maize and sugarcane, plasmodesmata have been observed which connect the cells of bundle sheath with adjacent mesophyll cells. Now it has some, that is the characteristic feature regarding the anatomy of these C4 plants. It is also called as the Kranz anatomy. Why it is called as the Kranz anatomy? Because the presence of the radially arranged cells of the bundle sheath, that radially arranged cell bundle sheath around the vascular bundles which look like a ring or wreath, because in the ring in Germany or in German language it is called as the Kranz. That's why this anatomy it is also called as the Kranz anatomy, because the bundle sheath which is surrounded by the vascular bundles, and that radially arranged bundle sheath surrounded to the vascular bundles which is looking as a wreath or a ring like structure, and in German it is called as the Kranz. That's why the name Kranz anatomy. So various steps of Hash-Slack pathway which involves, that is the two carboxylation reactions are taking place in this pathway. That is one take place in the chloroplast of mesophyll cells, and another carboxylation reaction takes place in the chloroplast of the cells of the bundle sheath. So here the first carboxylation reaction takes place in a mesophyll cells, the chloroplast of the mesophyll cells. So here the first CO2 acceptor is the phosphoenol pyruvate or the phosphoenol pyruvic acid. So first compound which is accept or fix the carbon dioxide, atmospheric carbon dioxide, and that phosphoenol pyruvic acid is made up of the three carbon compounds, and after fixing of this CO2 into a four carbon compound oxaloacetate or oxaloacetic acid, that reaction called as the carboxylation reaction. and here enzyme is the phosphoenol pyruvate carboxylase. This reaction is catalyzed by the enzymes phosphoenol pyruvate carboxylase and formation of the oxaloacetate or oxaloacetic acid. Now this oxaloacetic acid is reinterconverted or into a malate. That is, this is the dehydrogenation reaction, that is NADPH or converts to NADP plus, and again the 4 carbon compound, that is the malate or the malic acid is formed. So this malic acid, after formation of this malic acid in mesophyll cells of the in the mesophyll cells, in the chloroplast of mesophyll cells, the malic acid is transferred to the chloroplast of bundle sheath cells. Here, what happened? Again, the malic, here the decarboxylation reaction takes place. That is, malic acids are decarboxylated into a pyruvic acid, and simultaneously the carbon dioxide or CO2 is released here. So these CO2 are again accepted because here the Calvin cycles are continuing uh, in the chloroplast of bundle sheath cells here. And these in Calvin cycle, the ribulose bisphosphate are accept the carbon dioxide from this reaction, from the decarboxylation reaction to malic acid into pyruvic acid, and formation of here 3-phosphoglyceric acid, the first stable compound in Calvin cycle, in the presence of the enzymes, that is the ribulose bisphosphate carboxylase or Rubisco, and then formation of here that is uh, fructose, and again fructose it's converts into starch and sucrose, and remaining, that is 3-PGA 3-phosphoglyceric acids are converts into ribulose bisphosphate molecules and continue the cycle. So this is the second carboxylation reaction takes place in the Calvin cycle. Now remaining pyruvic acids are formed. These pyruvic acids are now ready to again back transfer to the chloroplast of the mesophyll cells here. This is the pyruvic acid. Now the pyruvic acids again here a phosphorylated here by phosphorylated by the phosphopyruvic kinase and enzymes are present. These enzymes are catalyze the reactions, and pyruvate are converts into phosphoenol pyruvate or phosphoenol pyruvic acid. And here again this phosphoenol pyruvate or phosphoenol pyruvic acid are ready to continue the C4 pathway or the Hatch and Slack pathway. Now again this phosphoenol pyruvic acids are ready to accept the another atmospheric carbon dioxide, and then again there is a start C4 pathway or Hatch and Slack pathway. Thank you very much.